couple of things. Relax. I'm not going to talk about my research. We're going to talk about stuff that's going on at NC State. The other thing is I want to tell Sam that if the National Science Foundation had a hit rate of 50%, we'd all be happy. It's down around 8 to 10%. So you're a great place to go to get some money, I'm telling you. So yeah, I'm going to talk about some of the challenges that we see sort of from an engineering perspective in terms of biorefineries and bioproducts, and some of the work that's going on at NC State that can have some applications, just sort of for a hint. So to start off, I wanted to sort of put out a couple of caveats. As best we can tell, the cost of biomass is always the dominant cost in any economic analysis, regardless of the product that you're looking at, regardless of the mix of products that you're looking at, regardless of the technology you're applying, okay? The cost of biomass is the predominant cost. And so anything that reduces the delivered cost of biomass is going to have a much larger impact on the operation of a biorefinery than if I can increase catalytic activity by 5% or 10% or 40%. And so, number one, cost of biomass. Number two, as any farmer knows and any forester knows, biomass is not biomass, okay? And it is the nature of that specific biomass. You know, Arundo donax is not, is not birch, is not loblolly pine, is not tobacco. So the nature of the biomass has a tremendous impact both on the cost and on the effectiveness of the processing of that material. Finally, fuel, sorry Sam, is a terrible product. And let me ask you this, right? Everybody drove here, right? Okay. Last time you filled up your tank, how much did you pay for gasoline? Too much, but per gallon. $2.05, $2.10, and you're happy about that, right? Because it's not $3. Okay, how much does a gallon of gasoline weigh? No, that's water. But that's a good place to start. About 6 pounds, 6.5 pounds. So you paid $2, okay, a little bit more for 6 pounds of fuel. So you paid 35 cents a pound, and you were happy with that. If it were $4 a gallon, you'd be screaming, okay? Which would be about 65 cents a pound. That's the problem with fuels. 40 to 50 cents a pound is what we're willing to tolerate on a regular basis, and we're ignoring other potential products from biomass, okay? And I'll just throw a couple up there because we collectively have experience with these kinds of things. What I'm going to be arguing, or hopefully persuading you of, is that what Jesse was talking about in terms of bioproducts is the way to drive a biofuels industry and not the other way around. If you base everything on the selling price of ethanol or whatever your fuel is, you're going to be constrained by this 30, 40, 50 cents a pound limit, practical limit. Whereas if you can make the kind of products that Titan, for example, is looking at, you can extract them or you can synthesize them from starting material and happen to make biomass-derived fuel on the side, you're going to be way ahead. So, the focus on ethanol was for a reason, but it's not really, oh, it hasn't started yet. I've got infinite amount of time. Thank you, Jacob. I'll start it for you. Spectacular. I think I just ate all of Jesse's time. So in thinking about, the sustainable bioeconomy and how engineers can have some impact on this, really what I'm going to be focusing on is sort of what happens during disassembly of biomass, and can we produce value-added chemicals from this, value-added products, and how would we go about doing that? And so let's, think about what is biomass that we're talking about. And so if we take lignocellulose, we're talking about lignin, we're talking about cellulose, we're talking about hemicellulose. And if you think about what you can do with these, almost everything goes through pretreatment. for starters, because we need to separate these macromolecules so that we can do something productive with them, because they are by nature significantly different. Lignin is an aromatic, and cellulose is a straight chain sugar, and they have very different properties, and you can do very different things with them. So it goes through pretreatment, and then what we're looking at are, what do you do with lignin? Lignin might be 25% of a tree's mass, dry mass, okay? For grasses, it might be more like 15 to 18%, but if we're talking about a lot of material, we're still talking about a reasonable amount of mass. So what can we do with lignin? And then cellulose, oops, go back. Cellulose, traditionally we have been looking at, let's hydrolyze it in some form or fashion, let's get sugars, let's do fermentation, let's make ethanol. And that's still fine, except that you run into that 50 cent a pound limitation. And so what I would propose is that the making of sugars is still a really good idea. It is the fermentation to more interesting products that probably is the future and is going to drive the development of this industry. And it is also going to be, not a return to let's make paper, but let's look at cellulose and hemicellulose to make higher performance fibers for extraction of heavy metals from drinking water, for high quality adhesives, or something else structural that takes advantage of some of the natural structure involved in these celluloses and hemicelluloses. Okay, all roads lead to pretreatment. Everything is, and that's because everything is affected by pretreatment. And so if you look at the research that's going on at NC State and everywhere, whether it's on conversions, depolymerization, hydrolysis and fermentation, or other modifications, everything revolves around the fact that pretreatment changes those molecules. Okay? Pretreatment has an effect on cellulose. It has a probably more severe effect on lignin. And so dealing with the effects of pretreatment, understanding what those are, is a really significant aspect in trying to accomplish any of these other objectives. And pretreatment comes back to pathway engineering, which comes back to the crosses and genetic modification, potentially, of crops, of trees, that will give that material the kind of properties that make it easier to do the kind of downstream processing that we need to do to generate these high-value products. Okay, I want to talk about one technology that is kind of old, at least in the pulp and paper industry, but it has a lot of really interesting applications. And that is refining. Refining is just a physical grinding, if you will, of woody biomass. Okay, or it can be any kind of lignocellulosic biomass, and we'll show that in a minute. But in the pulp and paper industry, you refine it, you grind it up to basically generate these little fibrils that can be sometimes single strands of cellulose, because it increases strength, it increases the interconnectivity of these fibers, so it changes the material properties, the performance properties of the cellulose that you have. And this just gives you an example of where some of the refining activities might be in either in a biorefinery that took green liquor from a pulp mill and did some processing to it to further open up the these fibers. And this is, like I said, this is relatively old, really old technology. And it has an interesting effect. One of the big challenges has been the yield of sugar from all sorts of different masses, and a lot of that has revolved around how can we separate lignin from cellulose. Well, here's a couple of examples where folks took some material that had 11% lignin, 15% lignin, 20% lignin, and put it through either a refiner for a certain amount of time or not, and then did enzymatic hydrolysis of the resulting cellulosic material. And what you see in each case is that there's reasonably good increase in the amount of sugar available and converted. And this is using C-Tech2 and H-Tech2, so I mean, this is not the latest and greatest, but the point is that that's what could be accomplished using that enzyme system, without refining, and then with refining, mechanical process, relatively inexpensive once you get past the capital investment, okay? Not onerous to operate. It's really well developed and well understood technology. Okay, so the challenge was, or the question was, how does it, bless you, how does it interact with other pretreatment technologies? All right, and so, another sort of study said, well, let's look at alkaline pretreatment, let's take hardwoods, and, let's look at what happens when we pre-treat this with refining. And this revolutions, it's sort of like, if you've ever seen inside an industrial bakery, the big dough mixers where you've got something that is spinning and it's also rotating around the bowl. Well, that's essentially what a PFI refiner looks like. It's toothed and cogged, and it rotates around this enclosure and chews things up, smashes it up against the side. So when it says PFI revolutions, this is 2,000 revolutions, 6,000 revolutions total. So it's a degree of treatment, if you will, all right? And what you see is that with a reasonable, couple thousand revolutions, you can achieve the kind of sugar yield with 5 FPUs of the mixture as you could untreated with 10. So, point being, you can use this kind of technology to reduce, Novozymes wants to sell you more, but it's expensive, relatively speaking, sometimes. And so, this is a way to potentially reduce the cost of and the need for enzyme use. Looked at dilute acid pretreatment and corn stover, okay, so, got away from trees, look at corn stover again, and saw that this is 2 and 4 FPUs, and sure enough, with modest amount of just refining, physical grinding of this material, you could achieve the same kind of sugar recovery with, again, sort of half the amount of enzyme. Now, obviously, it tops out. You can grind it so much, and then you're not getting the additional benefit. Autohydrolysis with sweet sorghum bagasse. Same sort of thing. Did the autohydrolysis at 170 degrees for 30, 45, and 60 minutes. Did enzymatic hydrolysis for, I think it was either 48 or 96 hours, and got the amount of sugar converted. And, again, what you see is that when you do some refining, you can see this 10, 15, potentially 20% increase in sugar yield. And in looking at all of this data, what it suggests is that when you're looking at low conversion, right, when you're looking at low conversion, any improvement that you see in conversion has to do more with the hydrolysis kinetics themselves. When you get to higher sugar yields, higher conversions, that increase is driven by the total amount of sugar available to be hydrolyzed, basically. And so the expectation is that what you're doing is you continue to open up the lignin cellulose mixture, if you will, or composite, I guess is probably a better description. you keep opening that up and making it more and more accessible to enzymatic hydrolysis. And if you look at the economics of this, and Jesse, you might have done some of this actually. Yeah, okay. I thought you might have. You should probably explain this slide then. What you see is just that there's relatively small charges for the additional electricity, the additional capital cost for implementing refining. If you add those up, that's 9 cents, 10 cents a gallon, which is $0.015 a pound. Okay. So not that significant. So it looks like this kind of refining enhances the digestibility by opening up the structure. It can be combined with a variety of chemical pretreatments, and if you use it with an acid pretreatment, it has the potential to reduce the amount of inhibitors that get generated, and so that helps you with your downstream fermentation. Okay, you're not generating as much inhibitor. This one's a little farther off the wall, okay? This is using electron beams, which is to say X-rays, with steam explosion and then enzymatic hydrolysis. And steam explosion, it softens the lignin, it increases the surface area, it helps open up hydrolysis. Electron beams, consider it a really, really, really strong dental X-ray, okay? And by that I mean a dental X-ray might expose you to about 3 millirem. This electron beam treatment that we're talking about is 100 million millirem. So yeah, it's a very fast suntan. And so the question is, what happens to the amount of sugar that we can get out with these pretreatments? And electron beam, like I said, it's a physical pretreatment. It tends to open up the amorphous and crystalline regions of cellulose. It breaks some bonds. It's high energy. It's generating in situ hydroxyl groups and hydrogen peroxide, so it's doing some local work with the water. And steam explosion, it's thermomechanical. It increases the accessibility of the material as well. And so they looked at the activity of, the effectiveness of electron beams and of steam explosion. They looked at, again, what the cost might be. And this is 75 kilograys, which is 7,500 million. Just to give you an idea about what the processing was, and I'm running out of time. This is regular dilute acid. This is E-beam and steam explosion. And what you see, the bio-oil is the E-beam steam explosion. And just what you see is that even with the, it only leads to a slight increase in in sugar yield, and it right now it doesn't look like it's incredibly effective. However, it's the first study that anybody's used with E-beams. It's not optimized. We're still trying to understand what it's actually doing and how it's actually working, okay? It's technology that's in the food industry now. Food is irradiated, okay? And so this is not totally exotic in terms of this can't be done. So this is a first look, and maybe for a first look, it's actually not terrible in terms of what they've been able to perform. One more question, and I'm in the purple, so I'm in flames, right? Okay. So I'm going to run through these last couple of slides. And it's actually not slide 50, well, it might be slide 57 here. My talk hasn't been that long. Another thing that we're looking at, that might seem like it, is can you advance biocatalytic systems to provide value added? And now, the value added can come from lignocellulose. It can come from oil crops as well, because, there's plenty of opportunities to do chemical conversions. I mean, that's really what this is all about. And so advanced biocatalytic systems could have a big effect there. Let me introduce you to a new concept, and then I'm going to have to run. And that is combining chemical catalysis and enzymatic catalysis. It's referred to as chemoenzymatic reaction systems. A particular example of this is the immobilization of enzymes on the surface of yeast. So the yeast make immobilized enzymes for you. You don't have to purify them. You just freeze dry the yeast. It's called yeast surface display. And yes, you can get 40 or 50,000 copies on the surface of yeast. We compared a bunch of activities with Novozyme 435. There was a publication years ago that tried to estimate the amount of protein enzyme in one of the beads, and so we tried to calculate to compare the milligrams of protein per gram dry weight of the catalyst, and there's about a 1,000-fold increase between our yeast system and Novozyme 435 as currently delivered. What I want to put in your mind is the fact that these enzymes can do a lot of interesting catalysis, and here's an example of combined chemistry where you do this allylation, you add double bonds to hydroxyl groups, and then you can create epoxides using enzymes, and it's effective. This is essentially, it looks like, gee, it only got to 50%. Well, it started to dimerize after that. The epoxide started to react. And so actually, all of the vanillic acid ended up being converted into epoxides. We see the same thing with organosolved lignin. Essentially, all of the theoretically available hydroxyl units are converted into epoxides, and then you start to see some reactions between them. So we think this may be a way of generating some epoxies. These kind of systems can work fine in non-aqueous situations, so any kind of chemistry you're thinking about, particularly since lipases are very good. You know, here's an example of taking the glycerol from biodiesel and making glycerol carbonate, which has very excellent solvent properties. Here's taking some straight chain fatty acids and making esters of various types. And given enough time, we can actually catch up with Novozyme 435, but there's no way we can catch them out of the gate. These kinds of systems are reusable over time, okay? And so they're actually fairly robust. And, you know, that brings us to the what's next. And everybody's seen The Graduate. The answer is plastics, which is to say a product, not a fuel, okay? Sugar yields, we're getting near the theoretical maximum. Supply chain efficiencies have been looked at very aggressively, but carbon usage is not, which is to say we've got to do something else with things that we consider to be waste. Okay? If we can get to total carbon utilization, then we're going to be making money hand over fist. But that's what it's going to take, which means we're going to have to develop catalysts. Those can be heterogeneous catalysts. Those can be new organisms, whole cells. They can be enzyme. We need to convert this underutilized carbon into a value-added product. And then repeat after me is we need to get past the first biorefinery in North Carolina, because the second one and the third one and the fourth one will be a heck of a lot easier to finance, a heck of a lot easier to get off the ground and going. Our big challenge is to get past number one. All right, thank you. It took a lot of people to do that work, including Jesse, who I managed not to list on there. And, and we've had a lot of support. Okay, so thank you for your attention.